Hello, I am Cassie Newman, and today I will be presenting my work performing an internal reference genome assembly, whole genome sequencing, and in silico digestion for improved efficiencies in marker discovery and genotyping in Virginia type peanuts. I am part of the NC State Peanut Breeding and Genetics group. We focus on the release of Virginia type peanut cultivars. The NC State breeding program has been based solely on phenotypic selection. However, DNA sequencing has become increasingly affordable, which has given breeders the opportunity to integrate genotypic information into their breeding programs. While there have been major strides in peanut genetics, these resources are not suitable for genotyping Virginia type peanuts. As shown in Figure 1, only 2 to 3% of the markers on the popular Affymetrix Arachis2 array are useful for genotyping Virginia type peanuts. Furthermore, as shown in Figure 2, there are other genotyping technologies that are cheaper than the array on a per-sample basis. Therefore, genomic resources specific to Virginia type peanuts must be developed so that we may exploit genotypic information to ultimately create better Virginia type cultivar releases. My work directly addresses this need. If you look to the right, I will describe my materials, methods, and corresponding results. The first step of marker identification was to assemble a Virginia type peanut genome, which will act as the reference for SNP calling. These SNPs will serve as genomic markers. The cultivar chosen to represent Virginia type peanut was Bailey 2 because it is representative of popular Virginia type cultivars and it has the high oleic trait for improved shelf life. The unscaffolded genome assembly results are shown in Table 1. Next, I obtained whole genome sequencing data from 96 lines, which are of interest to our breeding program. Figure 3 shows a breakdown of these lines based on prominent features. Figure 4 is a box plot showing depth of our first round of whole genome sequencing data. As a test for SNP identification, I used 9 Virginia type cultivars aligned to the Tifrunner reference genome. Figure 5 is a visualization of the resulting SNP calls on chromosome 1. Once I run the SNP identification pipeline with all 96 lines against the Bailey 2 genome, I will be able to optimize a reduced representation genotyping protocol. Reduced representation genotyping is essentially a pair of restriction enzymes that are used to fragment the genome, which reduces the amount of DNA you must sequence, also reducing cost. To optimize this genotyping protocol, an in silico digestion script was written which simulates the action of these restriction enzymes on the genome. This script allows the user to test these enzyme pairs in a matter of minutes. The output can be used to count fragments in a certain size range, as demonstrated in Figure 6, and you can also infer which SNPs would be captured by a particular enzyme pair, which is how I will optimize the genotyping protocol. Having presented preliminary results, the next steps towards the completion of this project are listed in the discussion section. To conclude, my work to identify markers and establish a genotyping protocol are the next steps towards integrating genomic tools into the NCSU PB&G breeding program. This work is the foundation for future implementation of marker-assisted selection and genomic selection. The application of these techniques will increase breeding efficiency for Virginia type peanuts. Thank you for viewing my presentation.